I want to make sure we dedicate some time to our audience questions that they've submitted. We have some really good ones here. And so I'm going to start with this one. And feel free to, among the three of you, jump in as you see fit. Are you considering logistics, for example, fit of the primary container and shipper's shipment temperature when you develop your storage and freezing process? I'll leave it up to any one of you to.
I think all three of us could answer that question collectively saying yes. You have to consider those things, right? I mean,
exactly.
The idea is not that you have one size fits all. I mean, it's great if you have a platform that you're comfortable with, but the application, the cell type, the dose, the route of administration, these are all things that you want to take into account. And we talked earlier about logistics. If you have a multi-center study, you want to be able to make sure that you're enabling each center to be able to run the same process, right? Same unit operations as they're preparing what will be administered to a patient. So you want to be able to simplify that, but you're going to tailor it to your needs. You don't want to force your therapy into a specific platform if you don't need to. So you want to be able to optimize it as you can. So yeah, I guess the answer would be
also thinking about once the clinical site receives it, when you're thinking about primary containers, I think that you don't want something that's so out of bounds that a clinical site doesn't know where to store it or how to store it or where it's going to fit, or that it doesn't go into conventional packaging once it's received there, whatever that is. So you really have to think about the whole journey in the logistics chain from your facility to cryopreservation through shipping and then to the clinical site, where usually it is stored for some period of time prior to administration. And so really understanding all the touch points along that process help you conceptualize how you work, how the logistics will work, and then how the clinical site is going to work with your product, and making sure that all of those pieces fit together. So yeah, completely agree.
Good. Okay. Moving on to the next question. For low volume CAR NK cell therapy manufacturing with constant program changes, what's the best approach for filling, visual inspection, and labeling before cryopreservation? Is it manual, semi-automatic, or more automated? Based on your experience, how do you balance efficiency and the flexibility needed for innovation? That's a good question.
Yeah, so I can take a stab at this, just focusing on the beginning of the question, low volume, assuming that I know what that means. I think that kind of gives the question or the answer a little bit of a bias, right? And I think that at least Pratik and I seem to agree early on this conversation that when you are in early phase of development, again, presumably low volume, if that's what the person who's asking the question means, I think that you need to be very careful with immediate investment in automation, for the sake of automation, and that not only can you just get away with, manual operations, but as Pratik mentioned earlier, in those early phase cases, manual operations can have advantages in allowing flexibility where you're still learning or you're getting feedback from regulatory agencies to change things. It might be easier if you have a manual process. So if I understood the question correctly, that would be my answer.
Yeah. I think the only thing I would add, Bruno, to what you mentioned, and again, maybe we're interpreting from the question itself, but if there's small volumes, and remember, when you fill a vial or whatever you're filling, but let's imagine it's a vial, you're filling it, and you have a small volume, the idea is the withdrawal volume that you want. So you fill something with 150 microliters, maybe you want 100 of those microliters. So you fill it with 150 so you can guarantee 100 out. There's the tapered vials that you can find, different vendors provide them. The idea is that your extraction volume from that vial is much closer to the actual fill volume. You can get most of it out. There's really no vial that I've seen that you can get 100% out, but you're going to get 98, 99 or something. So you want to be working with that type of framework from the beginning if you already know that there's some flexibility that you might need based on dosing. The other element is, just as Bruno pointed to, automated systems. If you're thinking about the user requirements to invest in an automated system, you want to make sure that you have this broad range covered. You don't want to invest in a filling piece of equipment for filling that only can do one volume at one speed. That's not going to help you if you want that flexibility. Then manual certainly is the way to go. And then as you fine tune what those requirements are, you can look for automation solutions that might meet that requirement. But the investment would happen later. You want to be able to have something that works, and manual is the way to do it, I think, initially, if you need that flexibility. So, Erin, I'm not sure if we answered the question entirely, but I think that's how we would approach it.
Wonderful, and that's what we're looking for. Is Kate back? Is she with us?
Yes, I am. Sorry about that.
Okay,
Tech difficulties.
great. Oh, no, perfect timing. I was just going to say, I think we lost Kate for a second, but glad to have you back. Okay, we are going to move on to the next audience question. Do you have any comments about developing closed systems versus vials in GMP production? And again, I would open it up to any one of our panelists to take a stab at it.
Well, I think some of this goes back to, oh, sorry, go ahead, Bruno.
No, you go, Kate. I'm not sure that I understood the question, so you go, please.
I mean, where I think we're sort of going is back to the earlier question on automation, and if you want to use sort of an automated closed system to be able to do vialing or whatever it is. And I think, again, there are advantages to that. I also think that if you're using a manual process, if you are in GMP, again, the question is, what's the cost and the investment of building that technology infrastructure when that may or may not be what your final product is when you get to a commercialization, point, or you get to a later stage clinical trial. So sometimes you do want to right time it, because I think we have all learned in this biotech environment that money is certainly not growing on trees. And I think that, really making sure that we are doing what is appropriate for our phase of development, where we may have process changes, and those process changes may or may not work with the automated systems that you're qualifying to be used in these facilities. So if it's something that you can use and you have access for, certainly reducing touch points can reduce likelihood for contamination. It can increase consistency. It can do all of those wonderful things. The other thing I think, though, is that a lot of these operators in GMP are highly skilled individuals, and there is often less variability than you would think in manual processes that are well trained. So I will just sort of leave people with that thought. It's up to you when the stage and the timing is appropriate to build in those closed systems, but there is a cost to it.
Sure, fair.
Maybe it's worth mentioning before we move on to another question, is that not all automated or closed systems are created equal. And the idea here is that, think about QC sampling. You may have a closed system, but in order for you to pull a QC sample, you have to break that sterile boundary. Well, that may be not the solution that you want, right? So maybe there's another separate solution that allows you to have a QC sample taken, but it's taken in a way that allows you to not expose the rest of the cells to the environment. And that might be the right solution for you. But again, this is an iterative process. I mean, if I interpret, Kate, with the way you described it, it's an iterative process that requires really thoughtful thinking about not just how you do the actual unit operation itself, but really end to end, all the sampling, all the testing, all those things around it, and the flexibility that you may need, especially early on.
Thank you both. Okay, moving on to our next question. What challenges do you experience in compiling and reviewing batch manufacturing and testing data coming from multiple nodes in the manufacturing supply chain so that the batch review and release can be done efficiently?
Maybe I can start us here, and then others can add to it. I mean, that maybe there's a technical element to that question, but I think it's also a quality element to that question. So oftentimes what you have to do if you're working with different parties, different sites, there's an element of making sure that you have a clear communication plan behind how information is shared. One thing that we've done before is we've created templates on what information we're looking for and shared that across. So it's a kind of common reporting template that allows us to be able to gather information that we need to see in order for us to go through our release process, and they have their own process. But we need those quality management systems to be playing nice with each other from one site to the other. So there's some elements that have to be, logistics that have to be defined up front. But then, of course, time is of the essence. These are cell therapies that are not sitting around and can sit around for weeks at a time. You've got to move quickly. I mean, you've got minutes and hours to be able to, release a batch once you have it prepared for a patient. So that's the idea. But that kind of synergy between end users, sites, and systems has to be defined up front. What are we looking for? What is the most critical to do? So this maybe goes back to that minimum testing that Kate had described earlier.
Yeah, I think that's exactly right. What's the minimum requirement? And also making sure you align on your TAT and making sure that sites understand that you understand that. If it's manufacturing, that may be weeks. If it's dose prep for a patient, it may be hours. And so making sure that everyone's aligned on what needs to happen, making sure that there is good communication. And I find that making sure that quality is aligned for exactly what they need to look for and exactly what they don't need to look for, if that makes sense. So I think to your point, what are the critical pieces that we need to be able to release? And, it's not an exercise in penmanship. It's not an exercise in who can have the longest forms, but it is an exercise in making sure we can safely release the product in a time that is sufficient to get that to the patient. So, minimum requirements is a really good thing to consider.